Hemophilus influenza. Presented by Medical Wizardry. Do you know? Hemophilus influenza is the first free-living organism, whose entire genome has been sequenced. And it is one of the typical parasite present in the respiratory mucosa. Let us see the basic characteristics of Hemophilus influenza. It is a gram-negative, non-motile, fastidious, capnophilic, facultative anaerobe. It is encapsulated and based on the fine structure of the capsule polysaccharide, it is subclassified into serovars named from A to F. Among those serovars, serovar B is the culprit who is causing the infections in most instances and hence called as HIB infections. It is also found to be pleomorphic, which means that they will have variable shapes and can be told as in coccobacilli. H. influenza is a facultative anaerobe, which means it can survive in presence of aerobic conditions also. Growth of the H. influenza requires growth factor 5 and 10. This is one of the main reasons for why we are using chocolate agar for culturing H. influenza, since factor 10 is nothing but hemin, which is found in the blood, which in turn is one of the major constituent of chocolate agar. The factor 10 is required for synthesis of enzymes that help in survival of H. influenza in aerobic conditions. The factor 5, which is none other than NAD or NADP. Bacteria like Staphylococcus aureus produce this factor, which is why H. influenza proliferate in the immediate vicinity of Staph aureus. This property is eminently known as satellite phenomenon. Apart from these media, H. influenza can also grow in Fildes agar and Leventhal medium. It has to be noted that H. influenza cannot grow in an isolated blood agar. Shall we look into the pathogenesis of this interesting culprit? The pathogenic nature of the H. influenza lies in its capsule. The capsule is made up of type B polyribosyl ribitol phosphate, which protects the organism from the phagocytosis of the host immune system. This explains us why the H. influenza infection is more common in children who lacks anti-capsular antibodies, even though protected by maternal antibodies for some period of time. This reason also adds up to the fact why that H. influenza infection is manifesting in the immunocompromised adult as secondary complication. The main enzyme which is involving in the pathogenicity, hand in hand with H. influenza, is IgA1 protease. The transmission of the infection is commonly by aerosols or droplet infections. However, invasive infectious spread through hematogenous route is also possible. With the basic idea of how H. influenza causes the problem, let us now dive deep into what problems it may cause. The most common clinical manifestations are meningitis, acute epiglottitis, pneumonia, septic arthritis, pericarditis, otitis media, sinusitis, purulent conjunctivitis. Osteomyelitis is most commonly and almost in all situations caused by Staphylococcus aureus, but in children H. influenza gets the title. Diagnosis of the H. influenza can be done with lab investigations and culture with cerebrospinal fluid as specimen. Satellitism exhibited by H. influenza is useful to narrow down the suspect. Staining using the standard stains like gram staining and identification of the bacteria based on the morphology will be useful. Latex agglutination gives us solid evidence of H. influenza infection. Latex agglutination test is done by mixing the sample, which can be either CSF or urine sample, with a suspension containing specific antibody-coated latex particles. When it is done, agglutination of the bacterial cells with the antibody happens, which is visible for the naked eye. When it comes to meet the bacteria face to face, we have two options. One is to prevent its pathogenesis with help of vaccines or to treat the infection with antibiotics. The vaccine option leaves us two types of vaccines, which are polysaccharide vaccine and other is conjugate vaccine. It was found that, since children less than 2 years of age impose no immunogenicity to the polysaccharide capsule and hence cannot be immunized with such kind of vaccines. However, when they are given vaccines which are conjugated to T-cell dependent antigens, the problem was resolved and they become immunogenic. Therapeutic options are mainly based on beta-lactam antibiotics. Ampicillins are advised in patients with HIB infections susceptible to ampicillin. Ceftriaxone is used in the condition otherwise. Rifampicin prophylaxis are also given in some instances. There is also a saying that polysaccharide vaccine conjugated to inactivated tetanus toxoid is also available in some places and are used as last booster dose.
Thank you for watching the lesson. Now it's your turn to talk. Answer the quick question. What are antigenic shift and antigenic drift? If you know, don't hesitate to answer in the comment box below. And don't forget to subscribe to Medical Wizardry, as more updates are on the way.
For more interesting contents like this, follow us on www.medicalwizardry.com.